I am at one of India's most well-known laboratories, the Center for Cellular and Molecular Biology in Hyderabad. And I have with me Dr. Vinay Nandikori. He is the director of CCMB, but more importantly, he is somebody who works on the tuberculosis bacterium. TB is almost omnipresent in India. Most people carry the TB infection. I have never had a TB infection which has shown up, but I can say for sure that I have carrying a latent infection of tuberculosis. India has an ambitious goal on tuberculosis, but we are still trying to understand the tuberculosis bug. It is a crafty microorganism which has existed alongside humans for ages. In this, we will try and understand the basic science of the tuberculosis organism, its interaction with the host, and our goals of trying to get rid of TB and how science can help. There are just about a dozen researchers in 1.4 billion population of India who work on the hard basic science of the TB bacterium, or mycobacterium to be precise, even though it is so common. Dr. Nandikori, thanks for permitting me into CCMB. It is an old haunt for me, and also to be able to speak about tuberculosis, which is something which has fascinated me and India, and we have a huge burden of tuberculosis. So let's start with some things which people will want to know. What is the kind of burden which India has on tuberculosis?
Approximately 1.5 million people die almost every year because of tuberculosis. India does have the highest burden of death as well as the cases across the world. They're in millions. Point is that India being a tropical country, the spread is also relatively easier because the droplets, it's an aerosolic spread. So droplets can hang in a bit, and the crowd is much higher. So that's why the spread happens relatively easier in a place like India. Our population is very high. We have pretty high incidence of tuberculosis. It's our problem. We need to deal with it.
Certainly. Now, there is also an idea from the government on trying to eliminate it soon. I understand you are a scientist and not a policy maker, so I'm not going to press you on the deadlines. But we do have the Prime Minister talking about an elimination program, and what is that?
The idea is basically when you're talking about TB, see, quite a lot of it is about, it's a treatable disease. We all know that. If we actually can treat all the patients who are getting tuberculosis in a systematic way, the idea, the ambitious goal is to eliminate the tuberculosis as a disease burden within India. Achievability depends on multiple things, and I really don't want to get into that aspect.
Sure, those are policy issues, those are issues about reaching the medicine to the right people, those are clinical issues, those are issues about community adaptations, and that is, I understand, beyond the realm of basic science. Tell me a little bit about the mycobacterium. How has it adapted, and what are its characteristics?
It's a very intelligent pathogen. It's been there in Egyptian mummies. You'll find it in Egyptian mummies. You'll find it in different levels. At some point of time, people who were getting tuberculosis in Europe, they were looking at it as a fashion statement even, because you become pale. And so that was looked at that way. Nevertheless, it's now almost more than 100 years since tuberculosis, Mycobacterium tuberculosis has been identified as the pathogen that causes TB. We think TB is only happens in the chest or pulmonary, what we call it. But let me tell you, tuberculosis as TB happens in almost every organ of the body, in the brain, in lymphoid tissues, in bone, in gut, in the eyes, even in reproductive organs. Tuberculosis is a very intelligent pathogen with very strong cell wall. It cannot, even on a surface, it doesn't die easily because of its cell wall. So that is one of the reason it can be infective even after being in the air, being in outside for long hours.
Now, we'll come to how it remains latent, but how and why do you think, you're a basic scientist, did the bacterium evolve such a strong cell wall which is so hard to penetrate?
It has, I don't know whether it has evolved or not, but it has a very strong cell wall, a multi-layered cell wall. Most of the bacteria have something called a peptidoglycan layer. In case of tuberculosis, in addition to the peptidoglycan layer, you have something called arabinogalactan layer, and then you have mycolic acid layer. So it's pretty thick cell wall, and it is so impermeable to quite a lot of things, including medicines. In fact, if you want to eliminate tuberculosis on a surface, the reagent that effectively works is a little bit phenolic in nature. Complete elimination of something on a surface. So it's a pathogen that has about 4,000 genes, but it has evolved itself to produce this waxy layer, and it can really it can continue to survive even in most hostile environments.
But why did it evolve like this? Because there has to be some evolutionary pressure for an organism to evolve such a strong defensive mechanism.
Humans are the host. So as you know, our human immunity is obviously going to work on eliminating any pathogen that comes along. And pathogen is obviously going to evolve to survive under those circumstances. You've seen that with COVID, and you will see that with Haemophilus influenza, you see that with everything. So what it does is that it, instead of, tuberculosis infects actually the immune cells, macrophages, and it survives there. It's an obvious survival strategy. Evolutions are directly to do with bacterial survival strategies versus ours to eliminate that. It's a fight. It's a tug of war. Eventually, both of us evolve. We deal with the infections better. They deal with us better. They can survive in our body quite comfortably, or at least try to survive within our body, given its hostile environment.
Now, let's come to. There was a point when TB was considered untreatable, and people were sent to these homes sanatoriums where they recovered through proper nutrition and sunlight and good air. To now, when we have drugs against TB, tell me a little bit about these drugs which are used against TB, and we will also come to what India gave to the world, which is DOTS. That we will talk separately.
So there are 4 drugs that are used for treating tuberculosis: isoniazid, rifampicin, pyrazinamide, ethambutol. These are the 4 which are used in a combination to avoid or to make sure that the drug resistance doesn't come that easily. They're not used individually. And these 4 drugs, they came a little later. It is true that people used to go to sanatoriums, but once the antibiotics they were discovered, and when this isoniazid and other things started becoming available, obviously you have a better way to treat the pathogen. So that is the reason why we basically are in a situation where we have a way to treat. Problem is, tuberculosis, is a long treatment. 6 months to 9 months, basically involves us to take that antibiotic on a daily basis in the morning, whatever may be the combination. And it is not easy to continue the compliance. You feel terrific in about 2 months. So then the compliance starts dropping. The problem with any kind of a compliance dropping is you will slowly and steadily evolve drug resistance. So currently the problem is we are having more and more drug resistant pathogen. So what is the drug resistance load as far as tuberculosis go? 15%.
15%.
15% is the drug resistance load. And we think it is only pulmonary. As I said, it is already in the extrapulmonary. Load, approximately 15%. So in 15% of the cases, you have drug resistance. In many cases, it is also extrapulmonary, but we give the same treatment. Right? We give the same treatment for a little longer for extrapulmonary tuberculosis. So the whole concept is that we also have to find better ways to treat. One would be a faster way to treat. The faster in the sense, if you can treat in 3 months with a particular combination, most probably compliance would be better. Evolution of drug resistance becomes less problematic. But it's a fight. As I said, we give drugs, they will have to find some ways to counter the things.
Now, these drugs which are given, these four combination of 4 drugs, and you spoke about multi-drug resistant tuberculosis. There is also something called the extreme drug resistance. Yes. Now, how is normal tuberculosis, multi-drug tuberculosis, and extreme tuberculosis different? And are multi-drug and extreme increasing?
So, the way the drug resistance is defined in tuberculosis is if you are resistant to one drug, no matter which one, it is called monodrug resistance. If you are resistant to 2 drugs, it is multi-drug resistance. And if you are resistant to 2 primary drugs plus one of the secondary drugs, many other secondary drugs are given, then it's called extremely drug resistant organism. And if you're resistant to all of them, then it will be considered totally drug resistant. If you are really going towards extreme drug resistance or to MDR, one of the next line of drugs are given only under those circumstances. They're called bedaquiline or delamide. So these are the drugs that are given to people who have MDR TB. So these evolutions of MDR is the way, this is how it is defined.
No, but why do we have to take these drugs for such a long time? Give me I don't want the clinical explanation. With you, I want the basic science explanation on how the drug enters the organism, how it kills, and why do we have to give? In my own family, I've seen patients being treated for 9 months, and it's very difficult because the drugs have side effects.
Okay, you take E. coli or any of the small bacterial infections that you get, you put it on a plate, it divides every 20 to 25 minutes. Okay, and by next day morning, you will see a colony, white pulp. And this colony has turned millions of bacteria. So means it is dividing very fast. So if you want to kill it, you need only 3 to 5 days treatment. Now contrast this with tuberculosis, it divides once in 20 to 24 hours. One bug becomes 2 in 20 to 24 hours. That is slow, very slow by any pathogen. And if you are having stress on it, it divides even slower. And if it is you're pushing too much antibiotic and putting a lot of drugs and all, eventually it reaches a point of basically latency. So eventually the tuberculosis biggest problem is for us from our point of view is it divides slow. Means if you're going to treat a bacterium that divides that slow, you'll have to be long term.
Meaning most of these drugs would act only to stop the division of the mycobacterium rather than killing the mycobacterium cell itself?
No, they do kill it. They do kill the mycobacterium, which is dividing bacterium. But a mycobacterium that starts dividing or slows down to such an extent, it is more difficult to kill. They're called persisters. So they continue to be there in any kind of like when you start treating, like in any kind of a thing, you start treating with antibiotic in vitro model. In vitro in the sense when you are doing it outside. You start killing the bacteria, but it reaches a point, some of them are not going to die. They're not resistant even. They're called persisters. So they're refractive. They found a way to be refractive, or they found a way to be not see the antibiotic around them. Right? So that is a problem. With tuberculosis, maybe problem with others too, but point is, these some of the times eventually slowly pushes it to a metabolically less active state. That is what is latency.
Now, how well do we understand the bug?
There are 4,000 genes. As of today, if you really look into it, maybe very good work has been done in about 1,000 odd. So really speaking, there are a lot more characterization, there are a lot more basic biology needs to be done on this pathogen to understand the functionality of many interesting things. Many basic aspects are not known. How does it combat oxidative stress? How does it go into the host and survive in such hostile environment? What are the secretory factors? What are the proteins that are thrown by the pathogen into the host? In what way they're helping? Right? It controls host signaling. How the host is responding to a pathogen is signaling, and it controls host signaling, it controls host expression, it controls host Splicing. Okay, splicing is basically means if you have a certain proteins are made and it changes the kind of proteins made by the host. So it has a capability to control many aspects of the host, and that is what makes this pathogen very intelligent, and ability to survive comes from that.
So it's a smart pathogen.
It is a smart pathogen.
And we have learned, in a way, to live with it, because many people don't get infected even though they carry the pathogen.
It's like this. When a pathogen enters our body, our immune system is going to start defending itself. So if an infected cell is there, our best is to kill it. If you can't kill it, you surround it. So that's precisely what granuloma is all about. Granuloma is a point where you have infected cells in the middle, surrounded by multiple layers of various immune cells and various other aspects, creating some kind of an structure which can be even identified in an X-ray. So the granuloma per se is essentially a situation where your immune system is reacting, but bacteria is not spreading. But if things are weak, things are become easy. At some point of time, bacteria can spread into other parts, other tissues, within the lung, and then it is situating granuloma. Otherwise, it can eliminate completely, in which case it is calcified granuloma. It is not eliminated, nor is it spreading. It's latency.
That's what they say, that most of us carry, and I'm repeating that again and again, and people should not feel bad if they get tuberculosis. It is treatable. Most of us carry it. They say immunocompromised people get tuberculosis sometime in their lifetimes.
It's not that immunocompromised people get tuberculosis, but when you're immunocompromised, you are prone to multiple diseases. Tuberculosis is one of them. You can also get streptococcus pneumoniae, and people can die of pneumonia itself. So when you are immunocompromised, you do not have a body defense working for you. So that can be for multiple reasons. It can be sometimes when you take an elective surgery, you may have to be given things to decrease your inflammation. That can also create problems. Sometimes you are weak, you're not eating well enough, and you're in an environment where you don't feel like eating, but you don't eat, whatever may be the reasons. Sometimes it is nutritional dependence or nutritional deprivation, or it could be simply other infections like HIV. Whatever may be the reason, eventually when you are weak, things are going to take opportunistic organisms can take advantage of that. It is one of them.
Now, let's come to the host, which is humans. In humans, how does the bug interact, and where does it hide? Because most of the time we hear tuberculosis as a pulmonary or a lung infection. You said rightly that it goes to many places, but in humans, where does it hide?
It starts off with your macrophages. It is taken up by our...
What are macrophages?
Macrophages, okay. In our body, there are two kinds of immune systems. Innate is basically the first line of defense. And then you have adaptive, which is B cells and T cells. One produces antibody. Others are the ones which kill the infected cells, called T cells. So macrophages, dendritic cells, some of these things are basically neutrophils. They are essentially the part of the innate immune system. Means whatever, our skin is part of the innate immune system. So once the pathogen enters our body, it is taken up by innate immune system normally, and what happens is it kind of fuses it, he makes it fragments of it and presents it to the adaptive immune system so that we recognize it. You're chopping it into pieces, some parts of it, proteins and all, and you're presenting it so that next time around I see this pathogen, my immune system is active. And that is what is antibody production in body, and all those things are detected.
That's what we saw in SARS-CoV-2 and COVID-19.
Yes. So these are two systems which are well-oiled machines, right? I mean, you have innate system. The moment pathogen is seen, it takes it up. It finds way to present it to the other things, and over a period of time, you get adaptive immune response. Tuberculosis is an interesting pathogen. It enters through the same system that is used by many pathogens, but it doesn't leave, it doesn't die there. It actually survives in so-called macrophages, or our innate immune cells. By not allowing the kind of a vacuole phagosome that is formed to fuse and get into fragments in a lysosomal compartment. So it's basically a pathogen that has found ways to survive in our immune cells.
So again, a very smart organism, but scientists are smarter than the organism. And I was always told during the SARS-CoV-2 or the COVID-19 pandemic that the SARS-CoV-2 virus may be smart, but humans are intelligent and we will find solutions. Why is it that with tuberculosis we have been struggling and struggling and struggling, and we are not finding suitable solutions, simple solutions?
We have solutions. We have drugs. We even have drugs to treat MDR TB. Right? I mean, it's not like we haven't found solutions. We have been finding solutions. Any particular pathogen that you start talking about, you have vaccines. People are working on it, and you have drugs that can treat the pathogen. We have that. It's not that we do not have solutions, but if pathogen gets to a point, eventually your drugs are going. I mean, it's going to become refractory to the drug treatment. There is a certain pool that survives, and that pool can find the right time to come back up. That's the reason. I mean, but it's not that we haven't been able to fight the pathogen, nor do we have no treatment. In fact, that may be the reason it is not really doesn't flag off that much.
No, certainly, tuberculosis causes far more deaths per minute in India than any other disease per se. Yet, sometimes as a community, we seem to ignore it till Prime Minister Narendra Modi has given this target to us. We won't go into the targets, but let's talk a little bit about, or if you had something to say, then maybe add, or let's talk a little bit about vaccines, because everybody who's born in India gets a BCG vaccine. So if you want to say something before the vaccine, please add. Otherwise, let's step into the zone of vaccines.
It is true that tuberculosis infection is very prevalent in India, but it is not so in Western world. And even in India, it seems to be more towards people who are more economically in a more compromised situation than people who are at a higher strata of income. That may be the reason why it doesn't get as much attention and visibility. Possible. I'm not sure. It's also true that it is not as much present in Western population, maybe because of population dynamics and the way people interact with each other. So that's why I said it is our problem. We have to deal with it. We can't expect somebody else to deal with it. We have to work on all three aspects, making possible vaccines. But BCG as a vaccine doesn't work. We know that already. I mean, it gives you some level of protection in children, but in the long term, BCG vaccination realistically does not provide major benefits in terms of stopping newer infections from coming in. So, in one sense, all of us are faced with tuberculosis. You yourself said that, we may have a lot of maybe latently infected, and it may be true also, because if you do Mantoux test, we get positives. Yes. So, point remains that we have seen the pathogen. What does vaccine do? Vaccine familiarizes you with the pathogen, right? So maybe vaccinations have the periods of vaccination. We have to make more effective vaccines by adding few other important factors to the existing BCG vaccine, so that your immune system is going to be told that, man, this is the one, right? And we have to do this more and more regularly. Maybe periodicity of the vaccine has to be figured out. So there is a research that's going on, and it's not my expertise area either.
Yeah, but the BCG vaccine has... Sure. The BCG vaccine has been around for such a long time, and we are still struggling to find another vaccine which is more effective than BCG, and we continue, even though, yes, in children and younger people, the BCG vaccine prevents tuberculosis, but in adults we see that it doesn't work. So why is it so difficult? In COVID-19, we made a vaccine in less than a year. You are at the Center for Cellular Molecular Biology. The kind of facilities you have here are unparalleled in India. Can you give us a vaccine by 2026? If you gave us a vaccine for SARS-CoV-2 in less than a year, and that's Bharat Biotech, which is a few kilometers away from here, can't you give us a vaccine against tuberculosis? And you're a basic science person. Find a solution, boss. We urgently need solutions.
Okay, if you know BCG is what we call as a live vaccine, live attenuated.
Attenuated.
Okay, live attenuated. It's not dead, it's live attenuated. They, one of the ways is take the BCG which is working, make it more effective. The efforts are on. I mean, it's not on. Again, I said I'm not a person who works in vaccine area.
I understand.
So if I were to be, I would basically make more efforts by putting more important some of the genes which are necessarily going to give you a better immune response into the BCG, and that is similar efforts are underway. You are giving a live vaccine and still we are not able to get protection. I mean, it's quite similar to TB. BCG is, I think, almost, 99% of it is very similar to tuberculosis, other than few changes here and there. So, despite that, we are not getting protection. So, what people are been trying to do is to put more and more things into the BCG to make it an effective vaccine. But it depends on immune memory, and it also depends on the fact that you may have already seen the pathogen. So, how you control a pathogen which hides? Sometimes you may not be able to. So these are the facets which are, I would say, confounding factors, which is not as simple as, okay, let's put, what is the vaccine? Vaccines are multiple kinds, okay? There is something called subunit vaccine where you put a protein which is on the surface, find out and familiarize your immune system. Vaccines are inactivated virus, that is what Bharat Biotech has done it, where they have inactivated the virus and let your immune system...
That is the Covaxin.
And then vaccines are where you put a spike protein into DNA backbone of adenovirus, and that's the DNA vaccine.
That's the Covishield.
So you have multiple kinds. In all these cases, this concept is simple. Make your immunity recognize the protein that is coming in. That doesn't seem to be working for us. So it is a more confounding than simple case of, inject the inactivated TB, for example.
Sure. See, but the burden is so high. Why can't we get more resources to scientists like you to make sure that we get the results which we require for tuberculosis, even though we have an NTB program globally with WHO, and the Prime Minister Narendra Modi has also been speaking about. But why can't we get more resources? Where is it? Are you not asking for those resources, or you are not getting? Where is the hurdle?
I don't think we have a problem of resources. At least people like me do not have it, so I cannot complain. So we have everything that we need as far as tuberculosis, doing the basic biology tuberculosis is concerned. We have around 100 odd tuberculosis, 120 odd people, different scientists, both clinical and basic and host-related work people there in India. Point is not about put resources and we get something out immediately. So you have to basically understand the pathogen properly, and I believe one of the ways is to work together as teams, and we need to start working in small teams to understand one aspect or other more deeply, and that's something we need to start making efforts on.
So you're saying we need more interdisciplinary and cross-boundary teams to find solutions for controlling TB?
First thing is clinicians and basic researchers working together and addressing problems in a systematic way that both has basic relevance, also has some kind of a clinical parameters are very well known. And the other thing is multiple groups coming together to look at both pathogen and at the host. So, but we have to work towards goals. If we have clearly defined goals, which are larger than my own research, most probably we'll be able to address the disease in a better way.
Well, the Prime Minister has given a very tight deadline and a goal, and I'm sure when the Prime Minister Narendra Modi gives the goal, he also gives the resources, and he also expects accountability at the end of it. I'm not going to push you too hard on that because that's way beyond your domain as a basic scientist. Again, coming back to the pathogen and host interaction. See, we discussed pathogen, we discussed the host. Now, there is also an interaction between the two happens. That is when the disease comes about. Now, how well do we understand that?
there is a lot of data that has come in the last 20-25 years. It's not something that it's not an easy pathogen to work. If you want to make a mutant in the pathogen, it's an effort. Again, for the same reason, once in 24 hours basically means a colony appears in 20 days. After 20 days, you'll think that the result is negative, you're going back to the bench. And from the basic point of view, recombination events are very slow in there. Now, with respect to why we can't achieve, we can achieve. In fact, DBT has launched this Dare to Irrad program where they are looking into 3 different aspects. One of them is can we use the drugs that are already available in the market, FDA-approved drugs that act on the host, but somewhere they actually decrease the load of tuberculosis. There are multiple host targets which can, if you have a drug that actually acts on it, decreases the load of tuberculosis, which is called host-directed therapy. And the second aspect is looking at what are these drugs, whether the drugs have the similar kind of distribution across the body. So because, the reality is we treat extrapulmonary tuberculosis with the same drugs that work for pulmonary tuberculosis. Are they going to these organelles with the same level? That is something we have to work on. The third aspect is looking at all the pathogen that is present in the population. We have patients from across India. We need to sequence those genomes and find out what kind of variations or what kind of things are there in the pathogen that is found in our population. As of today, maybe 200 to 300 sequences are available. All these programs require multiple groups to come together, and we are doing that. So it's not something that it's not like there is no effort is underway towards this. And most of them are essentially, as you said, as I was telling, is the DBT funded programs.
So within this, you're saying that the TB bacterium, mycobacterium, there is a lot of variability within that very smart bug, meaning something in Hyderabad could be very different from what is in Shillong or what could be in Jammu and Kashmir or Andaman Nicobar. Is that the case?
May not be. But recently I saw a manuscript. But may not be is the thing. But having said that, we don't know. Number one. Unless you don't know, you can't even say may not be or may be. So first get the information. That's important. Second aspect is recently I saw a manuscript that was presented by one of my students. This is not study in our lab at all. They found out that there is one mutation in one of the proteins is actually responsible for that particular pathogen to become more extrapulmonary. So such kind of correlations are single mutation correlations. So if you don't sequence enough, you wouldn't know. You don't have to have major variations. You need to have smaller variations. Identify those small variations and find out if they are contributing in some way or other to either something becoming extrapulmonary or something spreading more, something becoming a multi-drug resistance.
Is it also correct that growing the TB mycobacterium outside the human body is a challenge?
No, it is a challenge for leprae. Okay, but not for tuberculosis. We can grow. But what happens is quite a lot of times, let's say an organ has, let's say some of the, pulmonary is easy, let's not talk about it. So with some of the organs may have an extrapulmonary infection. But when you take it out, want to culture it, you may not be able to culture it. And that's called non-culturable. Doesn't mean that the tuberculosis is not there. So these kind of challenges are there with the clinical samples. Growing pathogen per se is easy. When you're doing in the lab, we can comfortably grow the pathogen. We need a BSL-3. People wear a bodysuit from top to bottom and go and work there. But we know how to grow the pathogen in the lab. In the sense, in a BSL-3 lab.
Sure.
And also, we know how to make mutations in the thing. Time-consuming, but doable things. So it's not difficult to grow, but when it comes to clinical samples, quite a lot of times you may suspect empirically it is tuberculosis, but you cannot culture it. You don't have a confirmation.
We haven't gone into the clinical aspects because that is a completely different domain, way beyond the capabilities of a basic scientist, because clinical research on tuberculosis, when you have a whole institute of the ICMR which does that, and most recently we had Dr. Soumya Swaminathan as the chief scientist of the World Health Organization, also a tuberculosis expert. Given the understanding we have today, how soon can we get a situation where the burden of tuberculosis in India is reduced to a very big way?
That's again, as I said, it is not anything to do with what we work on. It is to do with how well the existing systems are implemented, how well the drugs, DOTS programs, drugs are reaching the patient, how well the compliance happens. It's nothing to do with what I do in the
No, I'm talking about from the perspective of the understanding which we have with the current bacterium host. Is it feasible and is it possible to think of a situation where we can drastically reduce?
We can drastically reduce. We have to continue to work on understanding what are the better, not better drug targets for future, because it will evolve eventually. And we also have to understand if we can use host-directed therapy. Again, it is a way to combat the multi-drug resistance that might come up. When we are talking about, drastically decreasing, then you're talking about giving drugs to people, identifying and doing all those things. Then the resistance will become a bigger problem. So we have to continue to work on this to make sure that that doesn't become a huge problem going forward.
Sure. Now, within this whole setup, and I should also say India gave the solution with the directly observed treatment system on treating tuberculosis. And again, it's a clinical issue, so I'm not going into that domain with you. On basic science, are there other similar bugs which infect humans which have similar traits like tuberculosis?
Tuberculosis in itself seems to be a very amazing organism. I don't think there are, Leishman is also intracellular, but I wouldn't really talk about other pathogens. For me, this in itself is confounding enough. And I really would like to know every aspect of this pathogen. One is obviously, as I said, one is understand the pathogen itself well enough to be able to say, okay, these are the points that you need to hit. Even if not directly, what we call as an adjunct means in addition to the existing DOTS program, add one more drug that can in fact impact latency, for example. That's something that I would like to work on. And when it comes to host, what if I hit some of the host, as I again keep on repeating, because once you understand, then you can treat. Understanding is very important. 1,000 genes is not sufficient. Understand everything that happens, understand everything what happens when it goes into the host, and look at what kind of evolution of drug resistance happens. All these aspects doesn't fall in my own area, but something that we need to do collectively to really combat this pathogen.
So that was Dr. Vinay giving us an understanding of the massive burden we have on tuberculosis, but giving us a perspective not from the clinical side, but from the side of a basic research scientist. And he used a word, the TB bacterium is still confounding the scientists. Till we understand the TB bacterium a lot more, finding solutions may not be easy. But yes, India is working very hard to try and control the disease burden on tuberculosis. And yes, Prime Minister Narendra Modi has given a very stiff target. And for that to be achieved, basic scientists, clinicians, community level interventions, vaccine developers, and even people like me who communicate need to get the message across. But one small message for anybody who has TB, don't be afraid, it is treatable. My own family, very close family, got tuberculosis and got treated. And it's no longer a disease of the poor, it is affecting the affluent as well. And TB is treatable. We can reduce the burden and get solutions to bringing the TB burden to manageable levels. A difficult task, a tall order. We've been struggling to find a vaccine against tuberculosis for such a long time. I carry a scar of BCG. I'm sure you also must be carrying a scar of BCG. All children, all Indians carry a scar, but we still get TB in adults. It is India's problem. It is a problem which is sometimes called an orphan disease. But it may be orphan for the developed world, but it is an important disease for us, and we need to find solutions. And I'm sure, given the capabilities at the Center for Cellular Molecular Biology, working alongside with the immunology labs, the clinicians, the hospitals, and the community workers, sooner than later, we may get solution to controlling tuberculosis. From one of India's highly lauded, highly recognized, and a lab which delivers, from the Center for Cellular Molecular Biology, Pallav Bagla.